We are good to get started. Well, welcome everybody. My name is Anna. I'm going to be helping facilitate our chat today, but we're going to be learning about the career and life of a really cool person. My friend Izzy is joining us, who is a molecular laboratory technician at the Field Museum, which means, among other things, that Izzy works with DNA, and DNA is really cool. It's like the genetic code that makes us all who we are. So we're going to learn a lot about Izzy's and lots of cool things. And if you have any questions, you're welcome to type those on the comments, and then those will get to me and I'll be able to ask those to Izzy. So feel free to throw us your questions. Without further ado, hello, Izzy. Hi, how are you?
Hello, hi. I'm so excited. I'm so happy to be on one of these chats, and it's great to be able to talk to you guys through a virtual format. And yeah, I'm at the Field Museum today. I am in our DNA lab. This isn't just a green screen, so I can't wait to show you guys around and show you what we do in here.
Yeah, I think that'll be really cool to actually be able to like see that space and some
We're taking the laptop around. It's gonna be great.
I love a good laptop tour. So it's like kind of a first thing. Can you talk a little bit about like what is it that you do?
Yeah, it's pretty fun. So molecular lab technician is just a lot of fancy words for saying that I work in the Field Museum's in-house DNA lab, which is really cool. So if you don't know, all museums are research institutions behind the scenes. So not only do we have things on display, we have so many items in our collections that we're not able to display because we have so many. And we have scientists and graduate students working all the time behind the scenes that are studying these specimens and trying to learn more about their natural history, like where they came from, how old the species is, how they've evolved, how they're related to each other. And one of the ways that we can do that, we can understand those things, is we can look at them, we can kind of take them at face value and look at their traits and try to match them up that way. But a better way that we can do now is with DNA. So we can study the DNA of a plant or an animal. So humans aren't the only animals out there with DNA. So anything that's alive on our planet has its own genetic code. And what we can do in this lab is we take our specimen, we turn it into DNA, and we can do all of that DNA sequencing to read the DNA and analyze it in-house. So we do that all here start to finish.
That is so cool. And on the note of DNA, we did have some questions submitted from Miss Green's 6th grade class. They wanted to know what made you want to study DNA?
Yeah, that's a great question. Science in general for me was always something that I was really interested in as a kid. Like I was the kid that watched all the nature documentaries and like knew all the fun facts about animals. And I really liked science, but the only science career that I knew as a child was being a doctor, and that just really didn't appeal to me at all because I'm really squeamish. So when I was in high school, I was like, oh, maybe I'll do art. And then I was like, no, maybe I won't do art. I was in the marching band, doing all that nerdy stuff. But what really changed my career path was when I was able to take AP biology class in high school. And I took a psychology class at a college near me. And I learned that I really enjoyed learning about just basic biology and how all of these things tie in together. And when I was in college in my first year, I took a class called systematic biology, which is learning about science, but in the way of how we categorize who a species is based on their DNA and how they're related to each other and how we study evolution of different species over time. And once I took that class and kind of started thinking through that mindset, it was like all DNA all the time for me. So I really enjoy it.
That's really awesome. And I think this might have some connection to some of the things you just talked about. This class also wanted to know what gives you your spark, what makes you keep your studies, or what inspires you. And I just thought that was a really beautiful question.
Yeah, I love that question. And I think it's really important to think about those things right now when, everything is kind of shut down due to COVID and my job isn't the same as it always is. But I think if you're a scientist, you really just love learning and you love learning about new things and learning about how things work. So the best part about my job here at the Field Museum, which is so unique, is that I'm not stuck studying one thing. On a day-to-day, I can be studying birds, I can be studying plant DNA, I can be studying lichen and fungal DNA. And the best part is that I'm just surrounded by all these like really smart and amazing people who are able to share their day-to-day work with me and are like as passionate and excited about it as I am. So that always keeps me going, and there's always something new to learn, which is just part of being a scientist. So I also enjoy that too.
That is always really fun. We'll never know everything. There's always more to
No, I think the more you get into science, the more you realize that you start thinking that we know everything, and then when you end, you're like, we don't know anything. So that's kind of the joy of it.
It's really fun. And do you think you could talk a little bit too? I know this is like referenced in your bio, but kind of your journey, how you ended up where you are today from
yeah, I think that's really important.
bio to now.
Yeah, so like I said, I was kind of interested in science in high school, and I decided in college that I was going to go for a biology major. But like I said, I didn't really know where to go with that. I didn't want to be a doctor, but everybody that was in my biology class when I was a first year was pre-med, or they wanted to be doctors. And there's totally nothing wrong with that, but I think some solid advice is you definitely want to keep an open mind. I think most of my friends that started as pre-med kind of moved to a different area of science when they got more exposed to things. So you should definitely feel it out and see what you're interested in. And there's no problem with taking it slow and not knowing. And like, I certainly had no idea what I was going to do with my life in middle school and high school, and you shouldn't know because there's so much to explore. And see what you're passionate about. So when I was in college, I did biology, and then I took that, kind of evolution-based class, which I was really interested in. And the professor that taught that class had some openings in her research lab. And the school that I went to, you could work in those labs for credit, like, when you're an undergrad. And I was really nervous, like, oh, I don't know if I want to do it. I don't know if I'm going to be good enough. And I was really scared, but my mom made me send an email to her saying that I was interested in, being in the lab. And I just took it as I can learn more about this. And if I don't like it, I can always say that I tried, which I also think is a really important thing. So once I got in the lab, I realized I really like solving those problems, and I like the lab bench environment. So I did that for a couple of years. And then one summer I had an internship doing research at my college in the same lab. And then the most important thing that happened to me going into my senior year, I had a research internship at the Field Museum studying lichen DNA here. And when I was graduating from college, the job here was open, and I was like, oh, there's no way I'm getting this job. But because I kind of had my foot in the door when I was an intern and I enjoyed it so much and I made those connections, I was able to move out here for work. So that was really important. So that's kind of my career journey, if that makes sense.
I guess you must really liken it here.
I'm liking it. I have some lichens that I can show you guys.
Oh yeah.
If we want to get a little spiel about some projects that I've been working on.
I'm all for a spiel.
Okay, cool. So if you guys don't know what a lichen is, you've definitely seen them before. It's what's growing on this tree branch. And a lichen it's like a partnership, an organism that's made up of a fungus and an algae that live together, kind of like a little sandwich. So the fungus is the outer layer, and it provides the home for the algae that lives inside and makes sugar from the sun. So it gives them sugar kind of as rent for, living with the fungus. So this is just a branch that one of our students brought back for me for when he went hiking that has like a bunch of different species on it. But what's really fun about lichens is that they live all over the world. So I have a little packet here that was collected from one of our graduate students who went to Chile and Antarctica to collect this lichen that is the same genus. So it's a distant cousin as this one that we have on loan from Austria. So when I say they live everywhere, it's literally every continent, including Antarctica, which makes a really interesting, thing to study because they live everywhere and we can look at how they're all related to each other, if that makes sense.
That is really cool. And I'm thinking, is that the only thing that lives on every continent?
I mean, I'm not going to say yes because there's always exceptions, but it's
Yeah.
definitely like probably one of the only ones that can live kind of everywhere, like every harsh environment they kind of thrive in. They can live in rainforests, they can live in deserts, they can live in tundras. Um, they can live on our structures, like they even live on metal, some of them, you know, that's created by humans. So they're very adaptive and they like everywhere.
That's so cool.
Yeah.
We did have a question. Somebody was wondering, was there anyone in your life that helped you with your career?
Yes, definitely. And I think one of the great parts about working in this lab is we work with a lot of students, and it's really important when you're in science to have people who are mentors for you, who can tell you how it works. Because when I was growing up, you know, my parents went to college, but no one in my family was really like in academic science where you're doing like really lab bench science. So I really had no idea how anything worked. So in undergrad, so when I was in college, my advisor, whose lab I worked in, she took that mentorship role for me. And there were a lot of other professors at my college that were just, I really admired them and they were super great. And they kind of helped push me along. She was the one that told me to apply to this job. She's the one that was like, you should apply for that internship. And I was like, there's no way I'm getting it. So to have those people in your corner is so, so important. And for me, like being able to pay that forward with interns that we have here is really crucial as well. And there's plenty of support that we have in our building too. So our lab manager, he's like a great person to bounce off of for ideas. And yeah. So it's good to have those supporting roles outside of your family for sure.
Yeah, that's like an awesome web of supportive figures.
Yeah, I think I got really lucky too because in my college, I think in our biology department, there are more women faculty than men. So it felt kind of more approachable for me to be able to ask for that kind of mentorship. So to have that kind of cohort of professors or teachers, like even my science teacher in high school, like she was, you know, go for it, you know, take those classes. So yeah, to have that access is super important. Yeah.
That's awesome. So I can see in the background of the lab, there's just like a lot of exciting stuff. Oh, and there's just a question.
Good. Okay.
What's
Fine.
with all the equipment and stuff behind Izzy?
Yeah.
That's
So
the question.
we can spend hours going through what everything is in here, but I'll try my best to give a little overview so we don't spend too much time. So I'm gonna take us out of my bench. So I'll first start by saying what's really cool about our lab that I've never seen anywhere else, that really drives home that, hey, museums are research institutions, is that this whole lab has windows behind us. So this is the museum public floor. So people can come in and watch us work. And people will be like really concentrating on what you're doing. And it can be like a little scary if you're trying to work on something a little delicate. But so we have all these people watching us at all times, which is super fun. So it's kind of a half exhibit space, but you know, half actual working lab. So I can take you through. So the way that we sequence DNA, hold on, take you over here. So when I extract DNA from a lichen, I'll show you what a tube of DNA looks like, and you're probably gonna be a little disappointed because it's just that little clear liquid in that tube there. But it's in there, I promise. So what we have to do, the DNA is floating in the suspension and we can't see it because it's too small, you know, for the human eye to see. So we have a lot of different techniques that we can use to check the quality of our DNA, make sure it's there, make sure it's an okay concentration. So the first thing that we'll do, here, this is a little bench back here, and this is our gel bench. And if you are getting into lab science, you will definitely do a gel. You know, once you get even some high school kids will do it. And basically what we do to take a look at that DNA, we'll tag it with a little molecule that glows fluorescent under a black light. And we will literally run it through a little jello mold on an electric current because DNA also has a slightly negative charge. So the DNA will run through the gel and light up as it's running, so we can take a look at its quality and how much is there, if that makes sense. Does that make sense? Kind of.
It sounds kind of otherworldly and magical.
It's not. It all really seems like magic, and sometimes it also feels like magic because we have a lot of like finger crossing that things actually work, but it's all very straightforward. It's like following a recipe. And these back here are two DNA sequencers. So if anybody in your family has done a 23andMe DNA test when they're looking at their ancestry, what they're doing is they're taking the DNA that's coming out of your cheek, whatever, if they ask for a little cheek swab or your spit, and they're isolating your DNA and they're running it through a sequencer to try and look for those markers that have the most variation in your genome. And when we first started sequencing the human genome, sorry, I'm so sorry, this is like really filthy. We use this 3730 DNA analyzer. So this is like the workhorse of the original Human Genome Project. And this will only run, one gene for us at a time. So humans, our DNA is made up of base pairs. So we have ATGC, and that is what makes up our DNA, and that's what we read when we're looking at sequences. This machine can only do 600 at a time reliably, and our genome is 3 billion. So to sequence the human genome on that technology, there are like warehouses full of these, and it took, $1 billion in 10 years. But the way that the technology is moving, what we have over here, this is an Illumina platform DNA sequencer. You can see it's a lot smaller, first of all. And this will give us the equivalent output of a human genome in like 72 hours. And it's a lot cheaper. So those are kind of the two sequencing technologies we have on deck. And
do they still use the old one?
We do. Yeah, it's still really reliable. So when we're looking at, Kevin uses it a lot. And he's studying shark genetics. And what he's doing is basically trying to find cousins and different like close relatives of the same species of shark. So he knows exactly what section of the genome to look at that shows the most variation, and it's very accurate on that machine. So we still use it occasionally for those kinds of studies. But in the summer a lot too, we have a lot of interns that are trained on that machine. So
that's fun. And yeah. So you showed us that little like clear liquid with the DNA in it. And I know in museums, like activity stations, I've done like wheat germ DNA and strawberry DNA where you like mix all the stuff together.
Yeah.
So if you're doing lichen DNA, do you have to like take the lichen, smoosh it up, and like mix it with all that?
Yeah. So if any of you guys are interested in doing a DNA extraction at home, like Anna just said, you can Google a strawberry DNA extraction, or you can email us and I can send you an instruction kit. And what you're doing is basically the same thing that we do in the lab, but the chemicals are a lot more nasty. We're not using shampoo, so it doesn't smell good, and we're working at much smaller scales. So I don't have it in front of me, but what we have to do, so our DNA is inside of our cells, right? And fungi and plants specifically are really rigid, and they have those cell walls. So to break those cell walls to get a clean DNA extraction that's not going to be contaminated, what we have to do is another piece of our equipment back here. I'm running. This is a tank of liquid nitrogen. So what I do is we have little ceramic mortars and pestles, and I will dump some liquid nitrogen onto the lichen tissue that I want to extract, and that'll make it super cold so I can grind it up. And what you're doing when you grind it with the mortar and pestle, when it's cold like that, you're physically breaking those cell walls apart so you can get the DNA out. So yeah, it's like pretty metal.
That is really fun.
Yeah, so we can do that, or what else do I have here? I have these little chrome beads. And what you can do is you put these beads in this tube, and we have this expensive machine called a bead beater, which just holds the tubes and moves it back and forth really quickly to pulverize mostly like plant tissues. So those are some ways you can do it.
It reminds me of that thing, was it on the internet somewhere, where they were like, does it blend? And they just kept putting like different objects in a blender. I feel like
or like the hydraulic press thing. It's very satisfying once you get everything into a powder. I can't lie, that's one of my favorite things.
So much destruction.
Yes.
Miss Lowe's class is wondering, what is the most shocking thing you have seen in your research?
Shocking thing. I'm trying to think of what would be shocking for people. I think what maybe you wouldn't know is occasionally you'll read that we've discovered a new species of mouse, or a couple of years ago my lab manager who does sharks, he discovered a new species of shark and it's like a huge thing, whatever. But when we're studying lichens, how we previously identified species and categorized them, we look at them and we try to look at those differences under a microscope or something and see what they are. Like, tell me if you can see any differences between, I think they said there were 7 or 8 species on this branch. So I think what's very shocking about my day-to-day research is when we're studying things that are, less popular than a shark or a mammal, such as, all different kinds of plants and fungi, we're discovering new species every time we're running something on a sequencer. So that's pretty fun. I don't know, I can't think of any fun names that like our lichen team has thought of, because everything is still so new. Like, they aren't able to dive in and kind of separate those things out. It takes a lot of time, and I'm not good at that categorizing work. So maybe one day someone will, name a Pritzker Lab lichen from Antarctica or something.
That would be very fun. Somebody asked, do you get to work with other scientists in your work?
Yeah, that's a great question. And I know why you would ask that, because the lab is empty right now. But that's because of COVID. The only reason why we're empty right now is because we're kind of limited on what kind of staff can come in due to safety. But I would say the best part about this job, especially in the summer, so there's only 3 of us that are full-time staff, and in the summer we can have up to 40 people working in here, both in the front. So this is our front lab area, and we also have like a full back that's a lot bigger that people are working on, and they can be working on anything, seriously. And I remember when I was an intern, the thing that I was taking home that was the coolest thing to me is that every week that I was working in the lab, we had scientists coming in from different countries, and there would be like a dominant language of the week. Like, one week it was Portuguese, we had a bunch of Brazilian scientists in, and then another week it was Mandarin, because we have, all these scientists coming in from all over the world. So collaboration is a huge part of any scientist job, but I think what's unique about this lab here is we're the only DNA lab in the whole museum. So anybody who wants to study anything that's in our collections, and we have everything, has to come through these doors. So what's fun is that we, do get to work with all different sorts of people from all places working on every organism, except humans. We don't do humans in here.
Okay, so no, I was gonna ask, like, have you just ever run your DNA just for?
I wanted to. We kept joking that, we have the capacity to run our own like COVID tests in here if we really wanted to, because they're very simple. And one year my brother wanted a 23andMe kit, and I was like, just send me the money, I'll do it for you. But what's hard about human DNA, so like, I am so different genetically from this lichen that I would never do this, but I could like sneeze into the tube of DNA and it would not contaminate the sample, because we're looking for lichen DNA, we're not looking for human DNA. So to study human DNA, you need, a room with negative airflow. You need to suit up in your protective gear and just be really careful and mindful of how you're working. And we just like don't have the infrastructure for that here. But there is a human and also like an ancient DNA lab over at U Chicago that they're building. That's really cool to see, and it's super secure. So that work does get done, but we just can't do it in here, really.
That makes sense. So humans are just like a dangerous, not dangerous, but just a contaminant. The risk of
Oh
you,
yeah,
human contaminating and all the other humans that are sometimes in there.
yeah, definitely.
That makes sense. There's another question. Do you ever get to travel to other places?
That's a great question too. And I would say if any of you are interested in, research-based science, the best thing about this job is that once you have your project and you're set to go, you are traveling everywhere in the world on, your institution's dime. So they will pay you to go out and collect these things and bring them back for study. So a couple of my coworkers, I feel like everybody I know has been to Antarctica for collecting missions, and I'm like the only person who hasn't gone, and I'm a little upset about it. We have people traveling all the time. You know, if it's in the country, there are a lot of scientists that work in here that are working on mammals that'll just road trip down to Arizona, New Mexico. But there's so much international travel too. And another thing that's really cool is that we have meetings. So when scientists want to share what they're working on, the way that we do it is we pick a country that we all want to go to, and the organization that's running the meeting will set something up for everybody to come together and share their ideas. So when I was in college, the first one I went to was up in Edmonton in Canada, which was really fun. And then I went to one in Texas. And unfortunately, the first big one that I was supposed to go on with our field museum crew was postponed until next summer due to COVID, but it is in Bonito, Brazil, which just seems like a super beautiful place. So fingers crossed that I can do that. But yes, there is a lot of travel involved once you get involved as a student. I know one person I work with in the same year went to Chile, Australia, New Zealand, and then I think they were in not Sweden, maybe Norway for a meeting later on. And then they figured while they're in Europe, they'll just go to Germany and do some more collecting. Like, it's ridiculous. So there is a lot of travel if you're into that kind of thing. And it's, you know, adventurous and really fun too.
That sounds really magical. And I hope you get to go to Antarctica next time.
I know. Well, like, it's a really scary boat ride apparently. And like that freaks me out, but I still want to go one day because, I mean, tell me about it. I would just cry the whole time. I'd be so excited.
You know, gotta see those penguins. We've got time for one more question, and I think this is a very nice one to end on. Somebody asked, what do you love most about working at the Field Museum?
I think one of the coolest things, I already said, you know, we get to work with so many people from all over the world. And I think the best part is that everybody is just so passionate about what they're doing. So you can, you know, pre-COVID, when everybody's in the building, I'd go upstairs to run to the bathroom, and then you run into somebody in the hall who's so excited about something. And then an hour later, you're in somebody's office, like looking at some dinosaur fossils that haven't been described by, you know, a paper yet. So you can't take pictures because it's very secretive, but look at this new species of dinosaur. It's just like, it's such a wild place to work. And as a kid, I remember I went to the American Museum of Natural History in New York with my mom, and she told me that I said to her, like, I need to work here, which I don't remember, but I know that, you know, all I wanted was to basically be a professional nerd and work in a place like this. And so every day since then is just a dream come true. And I also love all of the outreach that I'm able to do, you know, and show people that a scientist just isn't an old, you know, dude at his desk in a lab coat. You know, we wear dresses, we love talking to people, we're outgoing, we have hobbies. So I think breaking those molds while being able to do this job is really fun too.
That is so wonderful.
You must know, you feel the same way. It's so great.
I do. It's very magical. Like, yeah, museums are very magical places to work for all those things you said. Like, everyone is just very excited to be there and excited to learn and excited to share.
Yeah.
That's why we're all here right now.
Definitely. And I can't wait until we're all together again. But in the meantime, it's great to have these chats and, you know, keep the outreach going.
Yeah, for sure. And there's a lot more. So all the students that are watching right now, this is like the first one in this semester. So there's going to be even more fun things to experience from the museum, from your screen. So we're really happy to be able to come to you in this way.
Yeah, I also remember saying that, you know, if anybody has any additional questions that they couldn't get in, I'm super happy to answer them. So you can just reach out to us and somehow those questions will get to me. So I'm looking forward to hearing any, if there are any more.
Yeah. That is wonderful. Thank you for doing that. Well, thank you everybody for joining us today. I hope you have a wonderful rest of your day and are excited about the DNA inside of us all and the world of science and how we can understand it. Yeah. Take care, everybody.
Bye. Take care.